Welcome, everyone. Hello, hello. We'll wait a few minutes for people to join and then we'll get started. Welcome, everyone. We will give it another minute and a half for people to join in. Feel free to say hello in the chat. Let us know where you're joining from. We've got Grayson from MIT. So Monique, you rock. Oh, I'm in California too, Mary. Emory, Gainesville, Virginia. I'm from Virginia. Boston, Virginia. Oh, this is amazing. People from all over. This makes me so happy. Oh, I hope you're safe in New York, Gustavo. It's crazy with the storm. Wow. It warms my heart to see people from all over. Oh, we've got someone from Kingston, Jamaica. All right, that's where our seminar speaker is from. So wonderful to see where everyone is joining from. Okay, we will go ahead and get started. So welcome, everyone. Like I said, it just warmed my heart to see all the places from around the world, United Arab Emirates, where people are joining from. It's great. So welcome, so glad to see or know that all of you are here, and thank you for joining us virtually. I know it's this week and month and year has just been a lot. So we really appreciate you sharing space with us today. I want to let you know that automated closed captioning is available, and to turn that on, if you look to the bottom right of your Zoom screen and you click the CC live transcript, there's an option to turn that on. But if you experience any issues accessing that, just message Black and Neuro directly in the chat. Before we begin, I would like to acknowledge that I am joining today from Ramaytush, Ohlone, and Muwekma lands. We're all joining this event from places around the world, so I invite you to take a moment to reflect on the lands that you're joining from, as well as your relationships to those lands and the Indigenous peoples who have previously and continue to inhabit them. So again, welcome, everyone. I'm so excited to introduce today's Black and Neuro seminar speaker, Dr. Monique Mendes. She's originally from Kingston, Jamaica, which we had some people in the chat mention they were from. She received her degree in biology from the University of Florida, and I know we had some people from Gainesville. Monique studies glial biology, and so she was drawn to the University of Rochester Neuroscience program because they had a program that had a strong focus on glial biology, which can be sometimes difficult to find because glia, we know, are ever important, but often overlooked. But she completed her PhD there in the lab of Dr. Ania Majewska, and was researching how specific glia, microglia, undergo a process called self-renewal. She successfully defended her thesis on July 31st, 2020, and moved on to do a postdoctoral fellowship here at Stanford in the lab of Dr. Mark Schnitzer. Today, Monique will be talking about the challenges and rewards that she faced as being the first Black woman to earn a PhD in neuroscience at her institution. So really, really looking forward to it. I know we'll all be able to hopefully learn something from her today. So without further ado, I will pass it off to Dr. Mendes. Thank you so much.
Okay, thank you so much, Brielle, for that introduction. And hello, everyone from California. Give me a moment as I try to share my screen here and let me know if everyone can see my presentation. Is that good?
Looks great.
Okay, perfect. Okay, good morning, everyone, and thank you all for being here. Thank you to the Black and Neuro organization for the invitation to speak today. And thank you to Brielle for that wonderful introduction. Today, my name is Monique Mendes. I'll introduce myself first. And today I'll be discussing the challenges and rewards as the first Black woman to earn a PhD in neuroscience at my institution. And feel free to contact me via Twitter or send me a message on my website if you have further questions or would like to talk more about my journey. So before I jump into a brief background to my journey, I'd like to give an outline of the things I plan to talk about today. So I'll give a background on how I got to a PhD in neuroscience. Then I'll talk about a brief overview of my thesis and the work I did in my PhD, the challenges and rewards I faced being the first Black woman to get a PhD at my institution, and the lessons I've learned along the way. In the sake of time, I'll give a brief overview of my current work at Stanford, and I'll leave some time for questions and comments towards the end of my talk. So first, I am a woman, I'm an immigrant, I am first generation, and I am a neuroscientist. I didn't appreciate all of these titles at first. When I started high school, I did everything in my power to conceal my rich, beautiful Jamaican accent. I stopped wearing my hair in braids. I never brought Jamaican food for packed lunch. But over time, I've learned to love all of these aspects and titles of myself. So as Brielle mentioned, I was born on a beautiful and small island named Jamaica in the Caribbean. And our motto is out of many, one people. I grew up in Kingston, which is at the south end of the island here. And I went to elementary school, middle school, and part of high school here in Kingston. So you can say that I have Jamaican roots and I was American grown. I attended an all-girls school in Jamaica, and at that time, I knew I enjoyed science and I loved history. I loved showing this picture because if you look at the view, we could see the mountains from class. It was just a beautiful campus. And here at this institution, Immaculate Conception High School in Jamaica, I was introduced to classical music and the violin. And I put in this quote, which is in Jamaican patois, which is, it essentially translates to, we are little, but we are strong. So we are little, but we talawa. And as I mentioned, this school was the foundation of where I am today. Not only was I introduced to science, but I had a rich experience with music and violin, and I loved music. If you asked 13-year-old me what I wanted to do when I grew up, I'd probably tell you I wanted to be a violinist. I devoted a number of years to violin, but I was not a prodigy. I loved it. I loved orchestra. And when I arrived in America in the early 2000s, I experienced my first example of racism. To be honest, I didn't even know what it was at first. I couldn't find a word to describe it until many years later. To give you a little bit of perspective, I moved from a what you would describe as a classist society in Jamaica that had its own issues, but embraced and promoted a motto of out of many, one people. The world of orchestra was where I first experienced this, and it's still so fresh in my mind when a parent helper asked me if I was here for orchestra on the first day of rehearsal, as if I shouldn't be there or if it was not possible. And these instances in conversations are now part of my reality. I excelled in high school, I excelled in orchestra, and was accepted to the University of Florida a couple of years after this. And there I majored in biology. I did what every student around was doing, and at the time I looked for a summer job and research opportunities. And a friend suggested I apply to be a research assistant in a neuroscience lab. So just to give you guys a little bit of perspective here, at this point, I have never met someone with a PhD before. I didn't know anything about neuroscience, and I've never set foot in a research lab except an advanced biology lab class I took in high school while dual enrolling at Broward College. So to say I was unprepared is an understatement. I didn't know what to expect, but something about bench science and asking a scientific question was so fulfilling. I heard about the McNair Scholars program through an email that went around the biology department, and I decided to apply. And for those who are unfamiliar with this program, the McNair Scholars program is a federally funded program that is present at about 151 institutions across the United States and Puerto Rico. And it is essentially created in memory of Dr. Ronald McNair, who passed away in the 90s. And it was prepared to prepare undergraduate students for pursuing a degree in a doctoral study degree, and involve them in research and other scholarly activities. And this program was everything for me. I was, for the first time, surrounded by a cohort of like-minded and passionate individuals who loved neuroscience. And actually that year, I presented my summer research at the annual biomedical conference for minority students in Texas. And I think in this specific moment, I decided that a career in research was for me. I decided that neuroscience was where I wanted to be and where I love to be. And as I mentioned before, I've always been drawn to science. I fell in love with neuroscience while working in a stroke lab at the University of Florida. And I had so many questions that I could not leave alone. I stayed late to finish an IHC experiment and to run behaviors. I made an effort to ask questions during lab meeting, and I thrived in this environment and decided to continue on my career in neuroscience at this point. So a lot of high schoolers and graduate students asked me, what did I major in? Did I have experience in neuroscience? When I was at University of Florida, I majored in biology. I took all the biology courses, the necessary math courses, and my first neuroscience course was actually in my graduate studies. So I did a lot of neuroscience research. I read a lot of neuroscience papers surrounding stroke, but my first graduate course was in neuroscience was during graduate school. And I graduated from the University of Florida and went on to my PhD in neuroscience. So I applied and was accepted to the University of Rochester Medical Center and the University of Florida. And I had to make the difficult decision. Did I want to attend Florida and continue working on my undergraduate project in stroke, where I was creating a name for myself, I published 3 or 4 papers while I was in an undergrad, Or do I want to move 2,000 miles away and challenge myself, get out of my comfort zone? I chose the latter. I joined a cohort of 6 in 2015, and here's a picture of us over the years, and I really like to look at this picture as we essentially age over the 4 years, and just having this cohort of like-minded individuals who were so passionate about science and loved neuroscience was so encouraging and helpful through grad school, and I really appreciated having these individuals in my cohort during that time. So I get asked all the time as well by students who are thinking about grad school, what is it like? And this comic by PhDcomics.com really sums it up. Sometimes you do a lot of napping and it's acceptable. There is constant adult supervision sometimes by your committee, by your advisor. You get cookies for lunch during seminar, and some of the most common activities is cutting and pasting and adding stuff to your lab notebook. There are no grades, but you are encouraged to play well with others, and crying sometimes is normal. And I really like to bring this up in my talk sometimes to students, just to give them a little bit of a perspective that grad school is sometimes a little bit like kindergarten. But although I agree with the PhD comics on their depiction, grad school was at times very lonely and isolating. It forced me to grow up fast at 21. And although you are so buried in your work, it was a worthwhile experience. As I mentioned, I was surrounded by individuals passionate about science, but one of the things that really was challenging for me, and I will go into a number of challenges later on in my talk, was the isolating portion of my PhD journey. Being the first Black woman, the first Black woman with a PhD at my institution, really left me with having no mentors that looked like me, no postdocs at my institution that were there to really go to and ask for advice, and really people that I think are going through the same challenges that I was going through at the moment. So I took the time in grad school to learn how to ask scientific questions, design studies, and perform experiments. And in 2017, I successfully proposed my thesis to a committee of experts. And at Rochester, in Dr. Ania Majewska's lab, I studied microglia. And microglia are regarded as the resident macrophages of the central nervous system. They're active surveillance in the brain, and using in vivo two-photon microscopy, we have captured microglia in real time. The dynamics of these cells in normal physiological conditions are constantly surveying the brain parenchyma, as you can see here. So microglia are here, and they are surveying the brain with their long, extensive processes, interacting with neurons and also interacting with microglial cells in their surroundings. Microglia are also known to respond to injury. And as seen here, in this video that was taken of an acute laser ablation injury in mice, you can see in real time as microglia move over the course of an hour towards the injury site, sending their processes in that direction to resolve the injury. In addition, diverse roles are emerging for microglia in the healthy brain, from sculpting developing neuronal circuits to guiding learning-associated plasticity. In fact, we can capture microglia-neuron interactions in vivo, and here I have an example of a microglia making putative contacts with spines in mice, with new roles of microglia emerging. There is deep interest in understanding microglial birth and maturation in the adult brain. And these videos were taken during my PhD, and this is from the Tremblay et al. 2010 paper from our lab. So with new roles of microglia emerging, there's a deep interest in understanding microglial birth and maturation in the adult brain. And most recently, there's been a lot of work to understand embryonic microglial development and maturation over time, but little work on adult microglia ontogeny. So this is where my work filled in and worked on. So this leads to the overall question of my thesis work. And this was, how do microglia self-renew in the adult brain in vivo? And we hypothesized that microglia are born by the division of these single surviving microglia that rapidly acquire these mature morphologies and functions in order to perform their physiological roles in the brain. And to tackle this these unanswered questions regarding microglial self-renew and maturation, we developed a chronic awake in vivo pharmacological depletion model of microglia. And we decided to approach this question in awake mice. So before I get started, I'd like to thank a number of individuals that helped bring this project together. And here are their faces and where they added to this project. And in the sake of time, I won't have enough time to go through all of the research and the questions and so on, but please feel free to check out our newly published paper in eLife, but I will give a quick summary of what we found. So essentially, we did long-term repetitive in vivo imaging that does not appear to result in microglial activation, and what we found was that microglial homeostasis is tightly regulated under basal conditions. Therefore, to explore self-renewal, we used a depletion model that depleted microglia over time. And as you can see here, after 7 days of depletion, we tracked microglia over time up to 14 and 30 days post repopulation. And here we have a quantitative analysis of this, showing that the number of microglia during this period, the average distance between the microglia, and we also looked at a P2Y12 knockout, which we hypothesized would be an essential to microglial repopulation. So you may be asking, where are these newly born microglia coming from? And what we found was that these newly born microglia were coming via residual cells in the visual cortex. And what we saw was that these doublet cells here were splitting off and moving away and repopulating the region. And we found that there was no difference between the P2Y12 knockout animals versus the controls. So this in vivo imaging experiment revealed a much slower time scale of division. So essentially, we went ahead and imaged every 4 hours for 24 hours during day 2 and day 3, which was shown to have a rapid increase in the number of microglia. And what we found was that we determined the rate of division at the peak of repopulation, and we mathematically modeled the repopulation we observed with the help of Matthew McCall and Zach Brem. And for the sake of time, I will just show you some of the characteristics that we observed during this 24-hour period, and I'm happy to answer questions about this during the question and answer section. So we saw a number of microglia that did not divide. We found rapidly dividing microglia that divided after 20 hours, as you can see here. We also observed secondary divisions that once they divided the first time, they went ahead and divided very shortly after that and acquired this equal cell-to-cell spacing. In addition to that, there were multiple behaviors of doublets that divided at different times, very close in proximity to each other. So, to summarize what I've talked about really quickly, these adult microglia repopulation is driven by stochastic rapid and continued individual division of remaining microglia, and these newly born microglia are structurally and functionally mature shortly after division occurs. And lastly, both microglia division and maturation appear to be independent of P2Y12 signaling. So, I have a lifetime of learning ahead. But on the day I presented this research to my committee and to a virtual audience in the middle of the pandemic, this day felt particularly special. And what does it mean to be first? Honestly, I feel a sense of responsibility to be first. I also feel like I have so much work to do, and I truly hope and wish I am not the last. So during grad school, in addition to the rewards I experienced of learning so much and being able to collaborate with some individuals, I also experienced a number of challenges during that period of 5.5 years. And one of the biggest things was that I stepped out of my comfort zone. As I mentioned earlier on in research, moving away from stroke and a field where I was creating a name for myself, I moved to microglia and understanding the role of microglia and microglial ontogeny and maturation. I also stepped out of my comfort zone by moving to one of the coldest places in the US. I saw snow for the first time in Rochester. And this picture was by a friend of mine, Holly, that we had probably the biggest snow in the 5 years that I was there. So I encourage you all who are thinking of going to graduate school to do the same. Take a class you would not usually sign up for. Take a rotation with a professor who's completely outside of your field and out of your comfort zone. I did the same. I went away from stroke. I went to an in vivo 2-photon microscopy lab. I've never done any sort of imaging. I've never studied glia, and I have no regrets. It was one of the most fulfilling experiences working in Ania Majewska's lab. So, in addition to stepping outside of my comfort zone, I had to tackle something known as the hidden curriculum. And a lot of you may be wondering, what is the hidden curriculum? And this refers to a collection of implicit academic, social, and cultural messages, unwritten rules, and unspoken expectations. It's essentially an unofficial norm, behavior, or values of the dominant culture context in which all teaching and learning is situated. And as I mentioned at the outset of my talk, I'm a first-generation college student. My parents received a high school education and were not equipped to guide me through the hidden curriculum. And when I say hidden curriculum, I mean, for example, I remember calling all of my professors for the first year I was there, Dr. Majewska, Dr. Olszewska, Dr. Fox, Dr. O'Banion, for a full year, while everyone else was on a first name basis. And this was just a cultural difference. I was so accustomed to saying Mr. and Mrs. and Doctor, but after observing my surroundings and understanding this, I transitioned to the first name basis. I realized about a year later that asking many questions during seminar was important for my own education, but also for others. I was following the data of others and learning to provide constructive criticism. And I remember sitting in seminar my first year, and there were a couple of students, very senior students, that were asking so many thoughtful questions, thought-provoking questions every single talk outside of their field. And I sat to myself and I said, I have no idea how they're doing this. Like, how can they ask questions at every single seminar? Like, how are they so shy? I didn't think that was something I could do. And over time, I moved myself to the front of the seminar. I sat in the front row. I took notes. I read up papers before the seminars came out so that I could be well versed in the new papers that are out on the field. And I asked questions. And this was really, really critical to me and my journey and learning to provide constructive criticism. Lastly, networking and advocating for myself and others. And I understand that I realized very early on that this was critical. This was critical to my development and to my experience. So anytime I went to the Society for Neuroscience conference, I would take the bus in the area. And if I went to a restaurant, I would talk to the person next to me that was wearing an ID. If I went to get coffee, I would introduce myself in the elevator. I tried my hardest to ask a question during those big talks at the Society for Neuroscience conference or any conference. And I always started off with, my name is Monique Mendes. I'm at the University of Rochester Medical Center, and I study microglia. And I was always excited to learn more about other people. Networking is a critical thing that I had to learn later on in my career. So I understand that these are a couple of examples, but these are just what could come to mind. And at the end of the talk, I'd love to hear your thoughts on this subject and how others have mastered this hidden curriculum. So this is another challenge that I experienced throughout my PhD career, and also as the first Black PhD neuroscientist at my institution. And I still struggle with imposter syndrome today. I find it comforting to attend talks like these and hear that other tenured and successful scientists have or are experiencing this imposter syndrome. And some of you may ask, what is imposter syndrome? And it's loosely defined as doubting your abilities and sometimes even feeling like a fraud. It's disproportionately affects high achieving people who find it difficult to accept their accomplishments. And I really like this image from Gail Gazelle, MD. And essentially on the left, it's showing you the assumption. So this is the assumption that many have. The yellow is what I know, and the teal is what I think others know. And as you can see, this is incredibly disproportionate and many times is 100% false. In reality, it's what others know and what I know is equally proportionate in your field. And I think this picture also really captures what a lot of people feel with imposter syndrome. And it's essentially someone asks, how are you doing? And you say, I'm doing fine. But at the back of your head, you're thinking, I don't know what I'm doing here. I just have no idea what is happening. And essentially, this was something I struggled with at the outset of my PhD. And one of the things and a piece of advice that I got from advisors and mentors during this time was that I do know what I'm doing. You do know what you're doing. You are here for a reason. And hearing those words from my advisor and from my mentors really assisted me to get to building my confidence and being able to accept my accomplishments over time during this time. I think one of the things that would have helped me during this period, though, was having representation. I think the only way we can move up above imposter syndrome is having individuals at different stages of your career that look like you, that are experiencing the things you're experiencing, that had to tackle and excel at the hidden curriculum and also just at science and in the bench. So this was something that I'm still working on. I still struggle with, but I am way better than I was at the beginning of my PhD career. So in addition to imposter syndrome, another challenge that I experienced being the first PhD in my institution in neuroscience was public speaking. I was painfully shy and I lacked confidence at the beginning of my graduate career. And luckily, you spend a lot of grad school giving talks. You spend a lot of time challenging others in public seminar, in your lab meetings, receiving criticism, giving criticism. And how did I transition from being so shy and not wanting to give a talk, and now? It honestly was practice. I stood in front of the mirror. I gave talks to my family. I went over my script multiple times to make sure that I felt comfortable with my project. And I also held on to a piece of advice from my advisor and also my other mentors, which is, no one knows your project as much as you do. You are the expert at your project doing these experiments on a daily basis. And at the end of each talk, I was always asking my advisor, Ania, how can I do better? What can I do? What did I do wrong? How could I have transitioned this portion? I live on feedback, and I was always open to receiving criticism about my public speaking, because there's always room for improvement. So I want to leave you in the last 10 to 11 minutes with some lessons I've learned and the rewards of my PhD career. So lesson number one that I learned very later on, about 3 or 4 years into my PhD, was to advocate for yourself. And I was many times the only Black person at a conference, the only Black woman at a conference, a seminar, in a class. And I embraced this fact. I sat in front, I introduced myself, I asked questions, I went to happy hours, I talked to people on the bus in random cities and at restaurants close to the conference. And you may be asking, what is self-advocacy? Well, self-advocacy is creating self-confidence, manifesting success, knowing your rights, being articulate, and being persistent. And before the age of science Twitter, I was doing this by going out and meeting other people at conferences and talking to them and reaching out to them. But I think right now, in this age where we have Twitter and we can interact with scientists real time, it is just amazing to see all of my colleagues advocating for each other, promoting each other's science, retweeting new papers and new opportunities. And I just absolutely love that. And on being the first Black woman to earn a PhD in economics and the first Black woman admitted to the Pennsylvania bar, Sadie Tanner Alexander, she said the following, and I love this quote: "I never looked for anybody to hold the door open for me. I knew well that the only way I could get that door open was to knock it down, because I knocked all of them down." And essentially what I want you to leave you with this first lesson is, you have to be annoying, but respectfully annoying. Persistency is key. The second lesson that I wanted to talk about that I learned during my PhD career was to celebrate the milestones. Take time, give yourself a pat on the back, share the good news with your science colleagues on Twitter, get a scoop of ice cream on the way home from lab to celebrate a solid Western blot, a successful master's exam, or just simply answering all your emails that day and tackling one thing on your to-do list. I was always going, and one day I decided to take a moment and enjoy the small victories. And I encourage you to do this often. Lesson number 3. You might be saying, I have a lot of people in my department and from conferences. Are those my mentors? How do I find mentors? Those people are great and helpful as you navigate grad school. However, I encourage you all to find mentors from all over at different career stages. And a mentor is an individual who lends his or her experience to advise someone starting out in a field. And I recruited a village. I'll go into that in a few slides. And I really appreciated Sally Ride's words here when she said, "It was a real honor for me to get to be the first woman astronaut. I think it's really important that young girls that are growing up today can see that women can be astronauts too. There have actually been a lot of women who are astronauts that's a career that's open to them. And that's how I feel. I feel that when you are the first, you feel such a responsibility that women can see, and Black women can see, that you can get a PhD in neuroscience, and people who have never even experienced or met someone with a PhD like myself can pursue a career like that. But this also involves finding mentors and having individuals in your corner. So here's a snapshot of my mentoring team and advocates. Does anything stand out to you? Dr. Samisha Barnes, who is a wonderful individual, was the first Black woman I ever met with a PhD in my career. And she was my McNair Scholars program advisor during that time. Dr. Sylvain Duray gave me my first lab research assistant position, and these other individuals were part of the collaborative team at the University of Florida and really advocated for me during this time for me to go to graduate school and to write and do research papers. Ania. Ania has been nothing but a mentor, an advocate, and on top of that, my advisor. She really challenged me to ask difficult questions. She advocated for me, and she was just all around an incredible mentor, providing opportunities for me and pushing me to be my best. I never want to forget Dr. Nathan, who was also the first Black person to receive a PhD in neuroscience at Rochester. And finally, this is the entirety of my mentoring team over the years. And each person over the many years, and even most recently my mentor now, has provided me with a different perspective, which has helped me mold me into the person that I am today and the neuroscientist that I plan to be as an independent investigator. And one thing that really stands out, and I'm hoping in the recent years, that I will have more individuals, more Black women neuroscientists on my team that will help me navigate the next steps of my career. So I realized that I wanted additional women on my team. So I asked Dr. Ann Etkin, a member of the Neuroscience Scholars Program, and she agreed. And she has been such an advocate and a mentor for me. And this is us meeting up in San Francisco most recently. Dr. Yasmin Hurd was the first Black woman neuroscientist I saw speak at the Society for Neuroscience conference in 2018 at a luncheon. And Dr. Michelle Jones-London was the second Black scientist I met in my career, Black woman scientist. And she is absolutely wonderful. She has been such an advocate for me and just someone who is so easy to talk to about different challenges I'm experiencing over my career. I say all of this to give you perspective. I attended 2 conferences a year, one specific to my field, and there was just such a lack of representation and mentors that look like me in my field. And this is why I am so happy for the Black in Neuro Initiative. This could not happen at a better time. I craved community and belonging in grad school, and now I have this in my postdoc. I remember when the website launched last year, for the first couple of days, I just refreshed the page and read all the bios and added everyone on Twitter. I was so excited. It was the highlight of my summer. I'm so excited to see what the Black in Neuro Initiative and Network has in store for the future. So most importantly, and most important lesson I've learned from mentors, and most important lesson I've learned from my advisor, Ania Majewska, is to embrace your failures as growth. And I repeat this to myself many times. I try to learn that to not take criticism personally, to learn that things that you apply for, grants, when you submit a paper, it's not a failure, but it's allowing you to grow and to learn more about yourself and to learn more about your research. So I keep this constantly thinking about this as I carry on in my career. I also appreciate Ania allowing me to take the time to teach and to advocate and mentor others. I made time to have undergraduate students because it was amazing to see them grow and to fall in love with science. And here I have two of my students who I mentored for one summer and then for 2 to 3 years. So what am I doing now? And I'll wrap up in the next 2 minutes, and I'm excited to hear your questions. I'm a postdoc at Stanford University right now, and a postdoc is essentially the next step on the academic career path after earning a PhD. And a postdoc is primarily a researcher who works under the supervision of a mentor as part of a larger research group. And as such, they conduct research, they either on a pre-specific project or one of their own design and publish that research. So right now in the Schnitzer lab, I'm using my in vivo imaging experience from my PhD to study astrocytes in learning and memory. And specifically, I'm interested in understanding how does neuromodulation through astrocytes regulate the stabilization of place codes and ensemble dynamics in a spatial memory task. And we hypothesized that the deletion of specific receptors on astrocytes will result in newly formed transient weak dendritic spines and place cell instability near the reward location in a goal-oriented spatial task, resulting in long-term memory deficits. So there's an evolving view of astrocytes. They were first described to be important in supporting the nervous system in disease. However, recent work demonstrates the role of astrocytes in sculpting neuronal circuits in the hippocampus. So to test this, we'll be using a custom-built 2-photon microscope to image spines and astrocyte interactions in mice and miniscope technology to identify changes in place cells near the reward zone. So overall, we expect in memory consolidation and retrieval as tested with, or we expect a deficit in memory consolidation and retrieval as tested with our spatial linear memory task. So I'll leave you with what about the future? My goal is to be an independent researcher at a research institution. I want to run my own lab. I want to do great science. I want to inspire and mentor young scientists. And most importantly, I want to continue learning and growing as a person. This is one of my favorite quotes ever. And this is from the first woman president of Liberia. And she said, "If your dreams do not scare you, they are not big enough." And with that, I'll conclude my story here. And I'd love to hear your experience and answer some of your questions. And before I jump into questions, I'd like to acknowledge my mentors and mentees, family and friends, the Black in Neuro initiative and network for all that they do, putting on these seminars and allowing us to talk about our experience. And finally, I'd like to thank the NIH DSPAN F99K00 Award, which allowed me to present my work, that funded my work. And with that, I'll take any questions and comments.
Thank you so much, Monique. That was just incredible. And there's lots of praise and in the chat and questions in the Q&A. So we'll go ahead and get to those now. Okay. Someone from an anonymous attendee, someone asked, how long did you wait after undergrad to apply to a PhD program? I think you talked about this, but just briefly, you want to talk about your trajectory.
Yeah, sure. So I graduated my undergraduate in 2015 of May, and I started grad school in July 2015. I had a month off.
So no break.
No breaks.
Okay, thank you. Our next question comes from Jennifer. She said, "Hi, Monique. Great talk. Love the insight. The hidden curriculum is something I've been struggling with in the field. What would you advise a pre-grad student to do in preparation for the hidden curriculum in grad school and in academia? Thanks.
That's a great question. Thank you so much, Jennifer. So I mentioned three things that I struggled with. I would say if there's anything I would tell myself maybe 3 or 4 years back to tackle the curriculum would be to find mentors early. Because essentially with this Black in Neuro initiative and the network, you have all of these individuals who are able to, you can contact them through the website, you can ask them for advice. Most students and postdocs are really excited and open to answering questions. But I would say get on science Twitter and find mentors as early as you can, and just ask them to just give them a little perspective on their experience.
Great, thank you. I love that you mentioned that, the hidden curriculum, because it is a thing that we all have to figure out along the way. And if someone would just tell us before we get there, it would be huge. I remember sitting in my first seminar and being like, how do these people know enough to ask questions and feel comfortable enough to do that? But like, you get used to it and you sit in front and you build up that confidence over time. And I don't know, if someone would have just pulled me aside and told me that was okay, it would have made a big difference. So I love it.
I know. I wish someone tapped me on the shoulder and said, hey, you can ask questions. It's okay if it's wrong.
And it's okay if you're a little nervous. Like everyone is. It's fine. I still get nervous. Okay. Our next question comes from Shraddha. They said, what an inspiring talk. I would love to hear more on how you manage to make time for self-care and creating time for your family, friends, and non-academic tasks. I've always known you as a very wholesome person, and I think it would be good to hear how one can fit all that in while being persistent in science. So some praise for you.
Thank you, Shraddha. That's so nice of you. So it's a work in progress. I will be completely honest. It's a work in progress. One thing I made time for during my PhD was orchestra. I made sure that every Wednesday evening at 6 p.m., my experiments, I would not have them run over because I wanted to make it to orchestra, which I went to the Brighton Symphony Orchestra, which was a community orchestra. I made sure that this was available time for myself because that allowed me to really just relax. It was kind of my relaxation. But I would say you really have to carve out that time. And if it means adding that time to your calendar, then do it. It's very, very important. It's okay to take a weekend off. It's okay to be off. And I know a lot of individuals feel like they must work 7 days a week, 365 days. I appreciated that Ania was very open. I went to school in Rochester. I had family in Florida. I would take 2 weeks to 1.5 weeks off to go home for Christmas, to go on a small vacation. It's okay to take that time. I think the first thing you have to do is to make sure that you take and carve out that time for yourself and add it to your calendar. It's very, very important.
Yeah, that's so important. And I would just like wholeheartedly agree with that. I feel like sometimes it can feel like you're adding another thing if you're like, this is a thing I'm going to do for self-care, but it's like a thing that takes time. But just like having that and meaning you have like a hard cutoff for work is just huge. So I love that you mentioned that. We had another question kind of about your trajectory. This one comes from Shaquisha. She asked, why did you decide to go straight through from your undergrad to PhD? And what might be the pros and cons of taking some time off and doing a gap year versus going straight through?
Ooh, that's a very good question. Thank you for talking about that. So, I debated taking a year off, and I almost took one off and went home to South Florida to stay at home. But my McNair advisor, Dr. Samisha Barnes, was adamant. She said, you're not taking a year off. You're going straight through. You're going to go and start your PhD because you can. Because that was another thing, too. I felt that imposter syndrome. I was like, I don't know what I'm doing. I'm going to start a PhD. I don't even know anything about science. or like doing like PhD science. But I think having Dr. Barnes push me to continue on was good. In terms of cons, I would say the first couple of weeks, I definitely had no idea what I was doing. I kind of just like went with the flow and like tried to see how people were doing things. I was very observant. But overall, I think the pros was that I had time to do an extra rotation because I started in the summer. So I did 4 rotations where I got to experience different techniques, work in different labs. So that was definitely a pro. And now I am on my way to my second year of postdoc. So I'm basically moving on, and that really helps getting to the next step. I would say if you feel ready and you want to start your PhD, go ahead. I think it's really up to the individual, but I decided to go ahead because I had Samisha, Dr. Barnes, really pushing me to go forward and believing in me. So,
all great points. It seems like there's pros and cons to both. You just have to figure out what works best for you.
Right.
So very good insights. We have a question from Stephanie. They said, I love this conversation. You're so inspiring. And I'm curious to hear more about what factors made you feel isolated. So kind of to connect with what you were saying about mentorship, are there things that previous mentors did or didn't do that helped kind of further perpetuate feelings of isolation or even alleviate some of those feelings of isolation?
Yes, all very, very good questions. So I think I felt most isolated in my first and second year of my PhD. And I think the real reason was because I didn't have mentors at that stage. I had my advisor. I didn't have my committee forming at that time. I really didn't know anyone. I was very new to the area. I had my cohort. We studied a couple times and met up, but I felt like I just was not really grasping, as I mentioned in my talk, the hidden curriculum, those little unspoken things that I felt really uncomfortable about. I was the only Black woman, at my institution, in class. I just felt a little bit isolated in those terms. And one of the things that really helped me around my second year was joining the Neuroscience Scholars program. And that's a program through the Society for Neuroscience. And I think that was the first time I met people that were like me, that were interested in neuroscience, that looked like me. It was amazing. I just had a cohort of individuals. And then I think the second time that I didn't feel so isolated was probably meeting my D-SPAN cohort virtually, which is the grant I received. In terms of what my mentors did and didn't do, I would say Ania, for example, was really a great advocate, and she persuaded me to apply for different opportunities and for me to go to France to study for a couple of weeks. So she really tried to push me to also be really involved at Rochester. So I was involved in a number of initiatives. So that really helped. I would say overall, I'm very, very happy with my mentoring team. The only drawback was that I didn't have that representation of a Black woman on my team and having that perspective. But I do feel that everyone was on in my corner and really pushing me upwards. So I hope that answered your question. I can answer after.
Yeah, I think so. Thank you. So we have so many questions in the chat. I'm going to pick one more, and then I'll save these so you can see them later if you want to try to answer them on Twitter. That's totally up to you. But we'll close with this last one. This is from Tanjiro. They said, hello, Monique. I love listening to your talk from Abu Dhabi in the United Arab Emirates. What advice do you have for a 10-year-old boy who wants to be a scientist?
Ooh.
Yeah.
That's a great question. Thank you, Tanjiro. And you're 10 years old. That's so wonderful. I would say I would say if you wanted to be a scientist right now, I would say read as many books as you can, ask as many questions as you can, keep asking questions, be inquisitive, and just create as many, like, have your goal of what scientist you'd like to be, and you'll achieve it. There's nothing that can hold you back at this point, because if you are thinking of being a scientist at 10 years old, then the sky's the limit for you. So keep asking questions. And thank you for asking a question here. That's wonderful. Thank you, Tanjiro.
I know he's already following your advice. Okay. All right. Well, unfortunately, we are at time. We have some additional great questions in the chat, but I'll send those to you privately afterwards. If you happen to have joined late and missed any of this, because there were so many gems today, or any of our other seminars, you can find them all on our YouTube channel, and I just dropped the link for that in the chat. Our next seminar speaker is a PhD candidate, Dr. Kelly Williford, and I'm already really looking forward to her talk. So we hope you'll check that out in our future seminars that are the first Friday of every month at the same time. Before we go, I want to thank my co-organizers, Ashley and Paige, and then Elena Dominguez today for help on the technical side of things. I want to thank the audience for joining us and sharing space. Like I said, it's been a crazy time, so I just appreciate so much for everyone that could be here today. Thank you for the incredible, insightful questions, and just everyone who joined us. As I've said before, kind of the goal of this series is to feature neuroscience faculty, neurologists, postdocs, and graduate students, and we want to provide a stage for them to share their story and their science, their setbacks and their triumphs. And we hope through this, we can really just like celebrate how wonderful and how resilient our community is. And I don't think we could have had anyone embody that better than Monique today. You were just wonderful. So the biggest thanks goes to you, Dr. Mendes. It was such an incredible privilege to hear about your story and your science, how you dealt with imposter syndrome, and how you've found your voice, and all the ways that you've just been like a game changer at every single level. So you're such an inspiration, and I'm just so grateful to be able to learn from you today. And I hope that everyone else was able to as well. So I want to thank everyone for coming, and I hope that you all have a wonderful day. Bye.
Thank you. Bye-bye, everyone.